Okay, let's look at the answer to Essay 5B. Or before that, let's look at the question. That is more important. The question asks you to explain the importance of specific sequences in the process of translation in eukaryotes. So, what's the boundary given to you, right? It's the process of translation in eukaryotes. Translation only, right? So there's no need to mention transcription, there's no need to mention post-transcriptional modification. This is only went on and on describing other processes that is not included in the question. Okay, so you also went on and on talking about how a protein is folded after translation. That's after translation, right? Not the process of translation. Right, so please again read the question, look for the boundary, then you focus on what the question is asking you for, right? And not waste time writing things that are not relevant. No matter how correct they are, they will not be awarded. Next, right? Next boundary or focus of the question is specific sequences, importance of specific sequences. Right, so you should be in your thinking process identifying what are the specific sequences, right? Give some description what they are, and then explain how these specific sequences are important during translation. So this is how you approach the question, right? And also read the answer in this in light of this, right? Read the answer for the specific sequences, okay, and how the explanation is written to explain how important these sequences are. Okay, then you give you a better understanding of the answer. Okay, so let's start off with the first stage of translation, which is translation preparation, meaning the addition of amino acid to the tRNA to form amino acyl tRNA. So this is the process, right? How are sequences, specific sequences, important for this process? So we can look at the specific amino acid sequence of the enzyme involved in this process, which is amino acyl tRNA synthetase, right? The specific amino acid sequence will give rise to the shape and therefore the function of the enzyme, right? So it's important, right? Importance of the amino acid sequence of the enzyme. Right, so again, the amino acid sequence gives rise to the shape of the enzyme, and the shape includes the shape of the active site. And we know that there are different amino acyl tRNA synthetase, right? And each of them are complementary to one amino acid and the corresponding tRNA. Okay, so how does the particular amino acyl tRNA synthetase recognize a particular amino acid and tRNA? It recognizes the shape of the amino acid via its active site, also the shape of the tRNA, right? And how does tRNA differ in terms of their shape? The difference is in the anticodon. That's where point 3 comes in, right? The specific anticodon sequence gives rise to a different shape of the anticodon of the tRNA, which is recognized by specific amino acyl tRNA synthetase through its active site. So with that, then a particular amino acyl tRNA synthetase can bind to a particular amino acid and bind to a particular tRNA and join the amino acid to the correct tRNA, forming the specific amino acyl tRNA. Okay, so that is how translation preparation is carried out with the involvement of specific sequences. Specific in sequences in terms of amino acid sequence of the enzyme and anticodon sequence of the tRNA. Okay, the next stage of translation is translation initiation. Right, so again, we are not describing the process, we are looking at how important sequences are. Okay, so firstly, we talk about the initiator tRNA binding to this small subunit. This one, no mention of sequence. Okay, but we need to talk about this because if you don't talk about this binding, right, then the point later, especially point 5 and 6, will not make sense. Okay, so still we need to describe a bit of the process. Okay, so that's step 1 or point 1. Then point 2 is where we go into the sequence. So we have the specific rRNA sequence in the mRNA binding site in the small subunit. And point 3, we also have the specific sequence of the 5' UTR on the mature mRNA. Mature mRNA because this is eukaryote, right? And we know that before this, there will be post-transcription modification that will have already happened. So the mRNA that we are talking about is a mature mRNA. Okay, so by right, I should be looking for mature, but then I did not. Okay, so just to let you know how you should improve in your answer, right, by being more specific to a eukaryotic context, and how I have been lenient, not being so straight up with some of these criteria that should have been mentioned in the answer. Okay, so anyway, back to the answer itself. Okay, so because you have the specific RNA sequence of the mRNA binding site, you also have the specific 5' UTR sequence on the mature mRNA that allows the small subunit to bind to the mature mRNA via complementary base pairing between the rRNA in the mRNA binding site and the 5' UTR sequence on the mature mRNA. So that's point 4. So you see how the specific sequences stated and the explanation of how they are important, right, in this binding during the initiation of translation. Okay, then now we have another sequence, the specific start codon sequence AUG, that will be required for the next stage or next step where the small subunit and the initiated tRNA moves downstream, and the specific sequence of the anticodon on the initiated tRNA will also be important to bind to the start codon via complementary base pairing to position the small subunit and the initiated tRNA at the start codon, which allows for the final step of translation initiation, right, the binding of the large subunit at the correct location where the start codon is to form the translation initiation complex. Right, so again, right, looking at the initiation process, it's not just a description of initiation, right? The focus is on how certain specific sequences, identified, named, described, right? How are they important, right, in this process of translation initiation? So I hope you see that in the answer. Okay, then for the elongation, okay, so for elongation, we require the template of the mature mRNA. And how is the template read? So the specific mRNA sequence of the mature mRNA is read in triplets that give us a specific sequence of codons, which determines the amino acid sequence of the polypeptide or protein translated from this mature mRNA. So that's the importance of the mRNA sequence, right, to be read as a sequence of codons. And the reading is 5' to 3'. That's point 2. And in point 3, right, the sequence of codons are important for complementary base pairing to anticodons of specific amino acid tRNA that will come into the A site. So that ensure only the correct amino acid tRNA enters the A site, bringing the correct amino acid to the ribosome for elongation. So that's the importance of the codon and anticodon sequence. Codon on the mature mRNA and anticodon on the amino acid tRNA to ensure the correct amino acid is brought to the ribosome. Okay, then we can mention the specific rRNA sequence that forms the peptidyl-transferase, which catalyzes formation of peptide bond to join the amino acids to form the polypeptide. So again, not just describing the elongation process, but highlighting what are the specific sequences required and how are they important for this process. Okay, next, termination. That's where we involve the specific sequence of the stop codon, and there are 3 sequences, UAA, UAG, UGA, which allows the entry of the release factor into the A site, hydrolyze to bring the water molecule to hydrolyze the bond holding the polypeptide and the tRNA in the P site. Note, it's not to hydrolyze the polypeptide full stop. Hydrolyze polypeptide means break down polypeptide, right? It's hydrolyze the bond between the polypeptide and the tRNA in the P site. Okay, so hydrolyze that to release the polypeptide, and of course the translation complex will dissociate. All right, so that would have gone through the 4 stages of translation. So you look at the process of translation, but we are not describing the process, right? We are looking at the process of translation and then identifying which specific sequence is important and then explain how they are important. So again, I want you to read the answer in that, in light of that, right, in that sequence, identifying the specific sequence, then explaining how it is important. I want you to read the answer like that, understand the answer like that, so that next time you can write like that if the question has similar requirements. Okay? Other points I accepted, right, would be a bit general kind of importance of sequence. So you mentioned the importance of amino acid sequences in certain proteins, namely the ribosomal proteins, in order to form the ribosome, right? The sequence of the translation factors or initiation factors so that they are functional, right? Sequence of the release factor so they are functional, sure, right? This is a bit vague, a bit more general, like just sequence, then therefore correct shape and function. Right? That's why it's only an extra point for me. It wasn't in my original answer, but I accepted it nonetheless. I think it's valid enough. And some of you also mentioned the specific RNA sequence, which will affect how the rRNA coils within itself, and also how the rRNA complex with the ribosomal proteins to form the ribosome or ribosomal subunits. Right? That's again quite vague, but still valid enough, so I accepted it as an extra point. So you mentioned the presence of the poly A tail. Although poly A tail is not really a specific sequence, it's just A's, right? So a better way to write would be specific length, right, of this chain of adenine nucleotides, which will then determine the half-life and the number of times the mRNA can be used for translation. Right, so a specific length for me is a better point, but I accepted you mentioning just the presence of poly A tail determining the half-life. Right, so I accepted that. Right, but really, if you think about it, and or rather to improve the answer, right, poly A tail really is not a specific sequence; it's just A's, right? So a specific length, right, would probably be better in terms of specific sequence. Okay, so specific length of poly A tail, then determining how long, right, it will stay in the cytoplasm and how many times it can be used for translation. Okay, so same as the previous essay, right? Every essay there will be 1 mark for quality of answer, and in this case, right, a bit lenient. I will give you the 1 mark if you included points from any 2 of the sections, right? Because there are 4 stages to translation, right? You should really talk about the different stages, right? So originally, I actually wanted you to mention at least 3 stages of translation, but never mind, let's make it more lenient, right? So any 2 stages, even the AVP, right? And as long as you have points in any 2 sections, right, in this answer, I'll give you the 1 mark already, right? So that to ensure some breadth in the answer. Okay, so there are a lot of comments, right, in the answer, right, as well as the previous answer for the 5A, right? I'm not going to go through the comments. Some of them I will have mentioned already as I talk about the answer itself, right? So please read through all the comments, learn from your own mistakes and also mistakes of others, so that you don't make the same mistakes again next time. Okay, so that's for the essays. Yeah.